Welcome to video 10 of Genetics, the Code Broken. In this video, we're going to be looking at mutations again and how they affect the genome, but we're also going to be looking at how we can actually try and repair those, or how our body works to try and repair those. So when our cells are dividing, the body actually has some processes in place that allows the DNA to be checked and repaired if necessary, to ensure that these mistakes aren't duplicated, and before this DNA is separated in mitosis. It's usually regulated by DNA repair genes, and repair of these damaged bases might occur by reversing the damage directly, or by excising, so actually taking out that damage and replacing the damaged base, which is currently in the DNA. The ability of the DNA to repair itself actually reduces the chance of perpetuating these mutations. So it means, making sure that these mutations aren't passed on to all of the cells in the body to maintain a healthy organism. So here we just have a quick graphic which I guess describes some of the way in which the DNA does repair itself. There's one where the DNA can be damaged by a single strand DNA break, and then there's also at the top we have the base excision repair. So it's where these repair enzymes come in to take out that the base that needs repairing and replacing it with normal DNA. Next, we move on to transposons, and these are pieces of DNA that can move from one position in the DNA to another. This causes rearranging in the neighboring segments of DNA, either by deletion, addition, or the relocation of some bases. There are, in fact, three classes of transposons. The first class are retrotransposons, and this is because they are involved in triggering the cell to make an RNA copy of their genes. This copy aligns with the DNA, forming a double-stranded molecule that can then be integrated into the chromosome. The second are class 2 transposons, and these are pieces of DNA that can move from place to place without freeing themselves completely from the chromosome. They are cut out from one position and inserted into another, and this can cause a mutation in the chromosome because it is in fact changing the structure of the of that DNA. Class 3 transposons are repeats of elements, and these are too small to behave as a gene themselves, but when they are inserted into DNA, it then again does cause a mutation. How, in fact, do these transposons impact on the genome? They destabilize the DNA in cells. They deregulate the reading of the genes. They can cause mutations through the chromosomal rearrangements, the point mutations, and the frame shift mutations that they cause. And they can, in fact, become active in bursts after periods of inactivity. And this can cause several characteristics to change at the same time, which can lead to inherited variations and the chance of improved adaptability. We now are going to go through two different types of mutations again, and be able to differentiate between germline and somatic mutations. So germline mutations are mutations that arrive in an organism's germ cells, otherwise known as gametes. If there are mutations in these gametes, these will be passed on to the offspring by sexual reproduction, therefore affecting the genome inherited by the offspring. The changes can be either favourable, unfavourable, or actually cause no effect, and they aren't expressed in the parent unless it directly affected the gamete production, and these play important roles in genetic diseases and in some cancers. The other type of mutation are somatic mutations, and these do not affect a species. In fact, they only produce a specific individual, as it can only be inherited by asexual reproduction or cloning. These only produce in a single organism's body cells during their lifetime, and it's not inherited from the parents, and it does not alter the gametes, and is not passed on by sexual reproduction. That's video 10. Make sure you tune in for video 11.